Today, our topic of interest is how scientists are using some of the body's own machinery to produce proteins or program cells directly within the human body to treat diseases.
Amazing. Who needs a bioreactor? I mean, we've spoken about how insect bodies are good bioreactors. I wonder if this is any different. The body itself as a bioreactor is what matters today on Discovery Matters.
Our first guest is our colleague, Scott Ripley. He's our resident guru of nucleic acids and genomic medicine.
And so how does Scott explain the idea of body as a bioreactor?
The bioprocessing space has long worked on how you can generate therapeutic proteins or other biological entities outside of the body, hijacking biological processes. It started off with recombinant proteins. You would have a classic cell biology-based approach using an upstream bioreactor, perhaps using CHO cells, HEK cells, or other cells which are grown in a cell culture media to express a protein, normally which has been generated through genetic modification of those cells, and then the subsequent purification of that. The body as a bioreactor starts to look at how scientists are considering using some of the body's own machinery together with advances in gene therapy, mRNA, and delivery technologies to actually start having the body express those proteins itself.
Body as a bioreactor is an alternative to expressing those proteins in an artificial way outside of the body.
Okay, so the traditional bioprocessing way is to use machines, as it were, to try and imitate what the body could do and then replace the protein of interest from outside of the body into the body. And now this new advanced medicines way is just to use the body instead of those machines, right?
Well, approximately, but we are not yet dumping tradition.
It's still very much the norm. What I've seen happening in the external world is the theory that you could hijack the body's machinery to generate a protein was always there. So it wasn't a could you, it was a why would you or why should you? And what we're starting to see now is very credible reasons why it is sensible to start considering the body as a bioreactor. Again, based on clinical need, manufacturing efficacy.
There's momentum building with some of the advances in the technologies and some of the reasons why you'd consider generating proteins inside the body, and that the body of science around
you didn't just do that.
is building all the time. I did, and I went all for it. The body of science around this is building all the time.
But is this proteins kind of like made by your body for your body, or can it just be for anybody or somebody?
Good question, and I asked that question to Scott by using the analogy of me and my partner Lars. You know Lars. So could proteins be generated in my body but used for Lars?
Ew.
This is using your own body to generate a biological medicine for yourself inside your own body. Now, going back to the Lars analogy, that could be a medicine that is specifically tailored to you. So it could be a personalized medicine that is designed only for your body. And we're starting to see that with some of the personalized cancer vaccines, for example. But it could also be a medicine that is given to everybody, but is produced singularly inside your own body and not shared with anybody else.
So Scott begins by saying that this concept was no longer a could you, but now a why should you? And how come we've got to this kind of logical step in medicine?
What you're starting to see is the conversion of 2 or 3 or 4 enabling factors all at the same time that start to make this possible. Very often, patient impact can be that some of the drugs are administered in rather large doses. If somebody takes a monoclonal antibody, it could be a large dose, it's a hospital admission, it's an intravenous drip to give them that. And some of that is because some of these monoclonal antibodies are cleared quite quickly from the patient's serum. So if you start to use the body as a bioreactor, then you get around some of the challenges of the half-life of the protein. Conceivably, a patient could go in, have one injection, and then the longevity of whether it's an mRNA or another type of entity inside the body could be expressing that monoclonal antibody inside the body for a longer period. I think that there's increasing evidence that some of the smaller antibody fragments or intracellular antibodies, which are very powerful entities that can start to reach some undruggable targets, their problem was always half-life inside the cell.
If you have the body as a bioreactor constantly expressing those intracellular antibodies, you start to get a much more powerful toolbox.
When you're working in an external bioreactor, you'd have one cell line expresses one protein. If you wanted to work with multiple proteins, you'd have multiple bioreactors and bring those together at the end of the process. If you start to use the body as a bioreactor, you can actually get much more creative in the number of proteins generated. There are examples out there now in the oncology space where perhaps 3, 4, 5, 6 different mRNA strands are introduced and they will express 3 or 4 different proteins. So you can have a multi-component protein system when you start to use the body as a bioreactor. It opens up a new toolbox in terms of different proteins that might have been cleared from the body quickly before, the ability to hijack the body's own machinery to work on dosing, the ability to get in complex protein combinations that perhaps would have been inefficient or uneconomical to make outside the body previously.
At the same time, not being able to isolate really complicates things and challenges the basic scientific need to be able to repeat experiments. That classic scientific law.
Yeah, and so that's going to be really quite challenging, I suppose. How does this concept work with quality assurance? How are we going to be able to monitor the process?
Obviously, you've got to look at safety for the patient. You've got to look at monitoring of the proteins that are expressed. There was a fantastic phrase thrown out by one of the scientific officers of one of the large mRNA companies that what is a bug in one system is a feature in another system. And it means they're starting to understand how to tune some of the characteristics of the expression inside the body. Do you want expression in a certain cell type? Do you want long-lived expression? Do you want short-lived expression? Do you want a burst of activity? Do you want a immunogenic response? Do you want a silencing response? So increasingly there is a toolbox that enables both the modification of the payload and through the lipid nanoparticle, perhaps the modification of where those are delivered to in the body, that lets you start to tailor some of those characteristics to exactly the quality that you're talking about. I think, look, that has to be matched to some of the individual genetic profiles of the patient.
And what about regulatory agencies? I mean, I can see them kind of looking at this and getting quite worried. How do they deal with it?
The way that I look at the creativity that's coming through in the nucleic acid space, is it likely that every single one of these mechanisms will make it to market? Certainly not. But what we start to see is this toolbox where it can be used as a vaccine. In the example of the COVID vaccines, it can be used as a recombinant vaccine. In the example of personalized cancer vaccines, it can be used to replace natural proteins. It can be used to introduce recombinant proteins. And that says to me that the base mechanisms of hijacking the body as a bioreactor, whether it's to express a vaccine or recombinant protein, there's going to be more and more understanding of the mechanisms. There's going to be more and more understanding of the analytics. And I think that in itself is going to help the regulators get comfortable with the behavior of some of these inside the body.
The breadth of the platform should mean that the regulators are going to understand how this is working in the body's machinery more and more because of the number of different applications, which are going to start to give some of that safety data and feedback data. Those are the normal guardrails that are put in place by the regulators, right?
Yeah, so the regulators need all of the safety data. So where exactly, in whose body are we testing it out?
We've seen the first biotech started up, which is there to deliver intracellular antibodies, but it is delivering those as an mRNA inside a lipid nanoparticle. So it's changing the paradigm. The old paradigm used to be discover an antibody, deliver an antibody, and the antibody works inside the body. Now it's discover an antibody or an antibody fragment, deliver an mRNA, but it's an antibody that is the drug substance inside the body. We've seen very heavy investment go into the cell therapy space, and a couple of examples there. One is using nucleic acid to deliver in vivo CAR T. This is moving into preclinical trials, into early trials.
So we're going to come back to the topic of in vivo CAR T later in this episode.
So is this like legitimizing biohacking, or is it? Are we a good way away from that?
I was going along the same lines during my conversation with Scott, and indeed, it starts to open up a very interesting moral toolbox.
I think you'd have the guardrails in place to cover those pieces. But in theory, what it does start to open up is if you get to the universal ability to produce perhaps an mRNA sequence and introduce that into an LNP, if you democratize that capability, anybody can sequence anything into that mRNA, and it starts to become a little bit simpler to introduce these. You could step away from perhaps some of the deep knowledge used to produce or manufacture a monoclonal antibody or an adeno-associated virus or others into that realm. But yeah, I think we're a long way from that.
So in the best case scenario, what now needs to happen for us to succeed safely for the benefit of these patients?
The next step is to watch these things progressing through clinical trials, and the whole thing is going to be demonstrating that this has a clinical benefit.
I'm seeing more and more relationships between protein discovery companies and mRNA delivery companies that I don't think there's activation energy needed at the front end to drive the science. It's going to require these things passing into clinic, getting the safety data, showing that there is a benefit to using the body as a bioreactor rather than the classical paradigm. But go back to the analogy, I think there are enough shots on goal at the moment that in the not too distant future we will see very legitimately the body being used as a bioreactor going forward. Now, it is never going to replace the classical paradigm. I think what we might start to see is perhaps some of the intractable proteins that are harder to express outside the body, niche proteins, smaller indications. That's of course where it is going to start.
This technology could also be a kind of science that would skip a generation.
The exact same way that the classic analogy with Africa jumping to mobile phones and skipping landlines. I'll give it by way of example. And a customer throughout, we were looking at expressing a CRISPR-Cas protein, and we thought, no, the CMC on that protein is going to be too expensive, so we just jumped straight to an mRNA LNP. And that's an example that the customer wasn't even ever expecting to express the protein. It wasn't a we tried to express the protein. They just went, no, let's skip a generation, let's go straight to mRNA LNP. So I think this is starting to happen right now. It's going to be the momentum of how quickly that builds at the moment.
Let's now speak with an expert who is harnessing the latest breakthroughs in CRISPR technology and genomic immunology to revolutionize how we understand and manipulate living cells.
I'm Justin Eyquem. I'm an assistant professor at University of California, San Francisco in the departments of medicine and microbiology and immunology. I also have a joint appointment with the Glasson Institute within the Genomic Immunology Institute. My lab is interested in improving therapies against mostly cancer. One way that my lab is particularly interested in is improving a specific subset of immune cell called the T cells, which we are doing it by reprogramming their genetic codes.
We're seeing more approvals from the FDA around CAR T, and Justin is in the thick of what's happening now.
Yeah, we were both on this call, which is a rare luxury to listen into one another's interviews. I just loved the way Justin described the focus of his lab's work.
My lab is really focused on improving CAR T cells, and there are several first incredible successes showing that CAR T can be highly effective against some hematological malignancies like in leukemia or in myeloma, and curing some patients that had no other options. But of course, as you mentioned, there are some limits. Even in patients that have been completely responding to these therapies due to either a downregulation of the receptor that the CAR is targeting or the lack of persistence of the CAR T cells, eventually the tumor is coming back. Another aspect is that while we are recently starting to see some signal in solid tumors, there hasn't been a lot of ideal targets to go after in solid tumors, which they would be expressed at the surface of the tumor cells at a high level, very homogeneous, and exclusively on tumor cells and not on healthy tissues that the CAR T could go after. And then the solid tumors are also surrounding themselves by what we call the tumor microenvironment, which is really hostile to CAR T cells and T cell functions in general. And one final aspect is the current CAR T cells manufacturing is really expensive, labor intensive, and takes a long time.
And since our topic of the day is body as a bioreactor, we asked Justin about his work with CAR T and how that relates to the topic, since, as we said earlier, traditional manufacturing of these cells happens ex vivo, so outside of the body in an actual bioreactor rather than a body bioreactor.
The whole field is really excited about that prospect because it would first just deliver a vector that brings the genes within the cells of interest, here the T cells within the body. So it would be called off the shelf, meaning that we don't have to take the cells for each patient, engineer them, and create new therapy. It would be one vector that you can directly inject, and it would be the same for all the patients for a given indication. Another benefit is currently when we are treating patients with CAR T cells, they need to have a lymphodepletion treatment where they're taking drugs that will deplete a lot of the immune cells to make space for the new CAR T cells to come and expand. Those come with toxicities, but also requires the patients to stay in the ICU because they would not have any immune cells for some short period of time. And so if now you're engineering the patients within the body, you can skip that lymphodepletion step, and which we think would be a benefit.
So with this annihilation of the immune system prior to the administration of CAR T, how often is that something that actually leads to complications and poor outcomes in CAR T therapy? I mean, I imagine it's a big risk.
Earlier clinical trials were tested without that preconditioning. The scientists in the clinical trials pretty quickly understood that that was essential, first to make space, but also they understood that by suppressing the immune system this way, simultaneously releasing some cytokines that were helping CAR T cells engrafting and proliferating. You're also depleting some cells that can have a suppressive activity on the CAR T cells, and it really showed that it was necessary. Then, yes, there are some toxicity associated with lymphodepletions, but it has not been really proven that some of those regimens can be associated with secondary malignancies, which is the resurgence of a malignancy later on.
So far, CAR T has largely been targeted at blood cancers, and now they're starting to see some promise in treating solid tumors.
And with that, I guess there's a series of key problems that need to be resolved to make the transition from blood-borne cancers to solid tumors, right?
Many labs are working in pursuit of identifying novel targets that would be expressed homogeneously within the tumor while not being expressed in healthy tissues.
There's a lot of DNA sequencing to try to identify mutations that might be specific to the tumors. And so the identifications of good target is really the key to have an effective therapy. Then there's the combinations of multiple targets to try to cover more tumors that might be a little bit more heterogeneous. Because of the rarity of great target in solid tumors, we start developing what we call Boolean AND gating. Can we program a T cells to not respond to a single signal, but to respond to a combination of signal? For example, AND gate, when you require a signal A and a signal B to activate the T cells, which means that you restrict T cell activations to tumor cells that we only express two antigens. And there's also OR gate, where T cells can recognize an antigen A or an antigen B. And there's a lot of different gating systems like this that are now developed to try to be effective while being precise and only targeting the tumors.
So there must be like particular ways or approaches or strategies that they're developing to enhance the function of T cells within the tumor microenvironment, because it's really harsh, that environment immediately around the tumor.
One approach is that the T cells is receiving a lot of immunosuppressive signal from that tumor microenvironment. There are strategies to create fusion proteins to rewire the negative signal into a positive signal. Instead of shutting the T cells down, the T cells will turn the negative signal and actually boost its function in response to it. Those are called switch receptors. And there are many flavors of those that can be adapted to any tumor type and type of immunosuppressive signal. We know that within the solid tumor, T cells are exposed to chronic signaling that can drive exhaustion or dysfunctions. And so there's a lot of work from my lab and the broader community that is trying to perform genome-wide screens to either systematically inactivate or overexpress some genes within a T cells and understand which of those genes we can modulate to perform for a much longer period of time.
Since there are so many options available to Justin and his lab, the goal is to understand how to use an optimal combination of antigens with receptors that rewire the immunosuppressive signal into a positive signal, and genetic modifications that make the T cells work better.
We would like to simultaneously inactivate 3 genes in the genomes and replace those genes by integrating others within them. This is what we call a knock-in, to integrate a transgene at the specific locations within the genome. We understand that if we want to achieve a response against all of those challenges, we're going to have to what we call multiplex, meaning doing multiple edits simultaneously. The problem is, since every time we do a modification in the cells, it's within the populations of cells that we're editing, it's never 100% of the edits. We have a mosaic of cells that if you try to do 3 knockouts and 3 knock-ins within a population of cells, at the end, there's only, out of all the possibilities, there's only 1 out of 64 possible.
Okay, so I get it. So while this could be more efficient, sort of doing 3 knock-ins and 3 knockouts at the same time, you actually multiply what you might call the number of error modes that you might have in any final cell during the modification. So like the good cells, you have a lot fewer of them in the final product compared to if you were just making one modification, I guess.
That's right. And Justin's paper, listeners, you can find Justin's paper in the show notes.
We're a proper podcast. We have show notes.
Provided the first demonstration that if you are optimizing your ability to engineer multiple loci, you can simultaneously use their developed methods to enrich those loci in one single step, which is necessary for large-scale manufacturing, where everybody wants to head to. Let's scale up, scale up, scale up.
So bringing this to the clinic, what's the next big challenge, according to Justin?
What my lab is really focused on right now is could we potentially bring those gene edits and could we deliver in vivo the machinery to edit the genomes but in a very precise way, so the T cells could then expand and recognize the tumor and kill it, really solving that manufacturing issue.
And this transition to in vivo requires a shift of the industry. Interest in vivo is increasing already.
There's already multiple biotech companies that have been created and large pharma that are interested in developing the vectors that will enable such technology. I think everyone understands the opportunity because it would solve a lot of the current issues that we have. But I think also the whole field understands the challenges that represent such an approach, because now you don't have the ability to do all the quality control on the cells that you do when it's ex vivo, which you can do before re-injecting into the patients.
Now, if everything is happening in the patient, you need to make sure that the vectors that you're developing are efficient enough to be an effective therapy, but also incredibly precise.
Yeah, you don't want those little targeted missiles going off in the wrong direction. The specificity, of the approach is key to the success of in vivo T-cell engineering. It just sounds so cool, doesn't it? I'm an in vivo T-cell engineer.
Yeah.
It's just like, who would have thought, 20 years ago that you could be that kind of thing? What about the challenges then and the benefits of developing and implementing these new in vivo therapies for patients? I mean, what are they going to bring over and above the ex vivo ones?
One of the most important aspects is the access patients. And that's why I'm excited about the in vivo, because it would, in theory, make the whole therapy easier and cheaper. If you think about the way the COVID vaccine was implemented at an enormous scale, if you could achieve something like that with the therapies that we're developing, I think it will be game-changing for the patients. The biggest challenge is can we do this extraordinary complex engineering while maintaining cost of goods to a level that it can be accessible to a very broad population? I think this is an incredible moment to be in this field right now, because those new tools are being developed and the whole field and industry is excited about it.
In review, today we discussed the avenues for leveraging the body's own machinery to produce proteins and program cells instead of ex vivo approaches. And that's why we called this episode Body as a Bioreactor. We explored that concept with insights from Scott Ripley, our own colleague, on how advances in gene therapy and mRNA delivery are making this possible. From Justin, we talked about improving CAR T therapies for cancer treatment by engineering T cells within the body. And you can find Justin's paper in our show notes. We delved into potential benefits, such as personalized and more effective treatments, and the challenges, which include quality control considerations from regulatory agencies, for example. So this technology could revolutionize medicine, making treatments more efficient and accessible. Connor, does that sum it up for you?
I think it does. And I think, as we watch this industry continue to go from, strength to strength, it's really extraordinarily exciting. Having seen sort of 10, 15 years ago, just the idea of being able to do this in a few people's minds. And now we're looking at this being a real reality going into clinical trial. It's absolutely astounding.
Let's keep learning, and let's talk about our every day as a school day.
Let's talk about what we've learned this week.
You first.
I was scrolling through my favorite science website, which is called theconversation.com. There's
I love The Conversation.
Well thought through reporting by scientific researchers and journalists. And there's a paper on there which focuses on the fact that the genes that birds carry shape their bird song even when they grow up far from home and potentially other birds that they might learn from. They recorded the songs of birds and compared them to the songs of their local Swedish environment where they were raised. So birds that were taken away from Sweden, and they then compared the bird song to them in their Swedish environment. And overall, the songs of the translocated males resembled the songs of those birds that stayed at home. And that came as no surprise because we know that social learning plays a crucial role in birdsong development for these birds, but they did discover that translocated males didn't just learn any old Swedish song, so they weren't just like popping out some ABBA moves there. They learned those elements that were most similar to a population aligned with their ancestral Dutch origins, which meant that their songs didn't completely overlap with the Swedish ones, but they resembled the Swedish and the Dutch songs. And that suggests that it's way back, and that these translocated males are genetically predisposed to learn songs from their ancestral population, which I guess should explain and manifest itself with lots of people in Minnesota singing ABBA. Maybe not.
Maybe you're right. It has something to do with our musicality, with the sounds of our languages, how easy it is for, some of us to learn the melody of a new language. I don't know. That sounds fascinating.
Yeah, it's brilliant. And you can see that genetic differences in a species affect actual cultural change. So our genes drive our culture. And we always think that those two, nurture and nature, are different, but they overlap so strongly. What about yourself? What have you learned this week?
Well, I'm doing more pleasure reading than I normally do. So I'm currently reading The Codebreaker by Walter Isaacson, which is the story of Jennifer Doudna. It is so good. So recommending to all of our listeners, pick up that book or listen to that book. It's excellent. But I started going down a rabbit hole with this book because early in the book, it's about Jennifer Doudna's childhood. She grew up in Hilo, Hawaii. And I thought, Hawaii, are there more scientists who grow up on Hawaii than the normal population? I went down and I found the National Academy's website, so the National Academy's Press, which says Hawaii is one of the best places in the world to study biological evolution. Why?
Oh, wow.
Because
it's so isolated.
Because it's isolated. That's exactly right. And native Hawaiian plants and animals are mostly endemic, which means they're found in Hawaii, nowhere else in the world. So you can study unique species. You have this enclosed environment where you can look at, the plant and animal species, compare them to other organisms. You can study volcanic activity. I mean, it's just a rich place to discover. Right? Yes. And so that was the childhood of Jennifer Doudna. So I was inspired, and perhaps I'll vacation on Hawaii one day and be able to experience that with my own eyes.
I think we might have to have a Discovery Matters field trip to Hawaii to just go and check whether this is actually the case. Our producer is Bethany Armit-Brewster, editing, mixing, and supervision by Banda Productions. Music from Epidemic Sound. My name is Conor McKechnie.
And I'm Dodie Axelson. Please do rate us, and rate us well, on Spotify or whichever platform you listen on. And we will be speaking with you when we come back with another episode of Discovery Matters.
Ta-ra for now.